capacity to activate and/or manipulate brain cells might give us powerful new techniques to investigate and cure neurological problems, and we've seen some really intriguing ways to do so. University College London researchers have proven in rats a novel sort of micromagnet that may act as a miniature mechanical switch to turn on touch-sensitive cells, paving the way for a new means that regulates specific brain areas. There are parallels between this accomplishment and optogenetics, a fascinating field of science that focuses on the precise regulation of cells in the body. This method entails putting genes into normal cells to make them light sensitive, allowing them to be activated to treat paralysis, pain relief, and eyesight restoration. Although there have been some success, such as case last year in which optogenic was used for the first time in a person for practical eyesight restoration, adopting the technique for clinical application has been problematic because of the necessity for a genetic alteration of the cells. UCL researchers have devised a new method of manipulating brain activity that does not require such metrics. Our novel technique employs magnetic particles and magnets for regulate brain cells activity remotely and accurately, and it does so introducing no device or foreign gene into the brain, says the researcher. Dr. Yichao Yu, the primary researcher, said that the team's magnet-centric strategy focuses on astrocytes which dwell between the brain blood vessels and nerve cells and provide metabolic and structural support to neurons while also controlling neural circuit activity. They are also touch-sensitive, which is convenient for the scientist because astrocytes are sensitive to touch. Coating them with the magnet particular allows you to prod them with a magnet from outside the body and therefore regulate their function, says the researcher. Professor Mark Lithgow, a senior author, said the ability to regulate astrocytes remotely gives a new tool for studying their function and may have the potential to treat brain and mood diseases such as depression, says the researcher. The team method is known as the magnetomagnetical stimulation, and it entails of use particles that have been coated with an antibody. They attach preferentially to astrocytes as shown by injections into specific brain areas in rats, and may then be turned on by placing a magnetic device near the skull outside the body. This triggered a cascade of signaling channels that are thought to control a wide range of brain activities. The researchers write the ability to control brain astrocytes while using a magnet field, providing researchers with a new tool to study the function of these cells in the health and disease, which could be important for the future development of novel and effective treatment for some common neurological disorders like epilepsy and stroke, the researcher said. The magnetic particles light up on MRI images, allowing the scientists to pinpoint their exact position in the brain. This, along with the technology non-invasive nature, has experts excited about the potential. We are very excited about this technique because of its therapeutic potential, says researcher Lithgow. Unlike previous approaches, MMS makes use of some brain cells' exceptional sensitivity to touch, so no genic change of device implantation is required. Thank you very much for watching. Subscribe for future videos.